Hi, everyone. This is Physical Principles of Quantum Biology, Part 2, Chapter 9, Electromagnetic Oscillations in Biostructures. This is, of course, a chapter from my new book with my wife, Brandy, Physical Principles of Quantum Biology. You can download yours from the archive or pick it up on any number of booksellers online. The digital object identifier is here on this slide with the archive link. As I mentioned, today we'll be studying Chapter 9, Electromagnetic Oscillations in Biostructures. And I, for one, am excited about this chapter because we're really going to get into some of the questions of where quantum coherence could play a significant role in cellular physiology. So let's take a look, shall we? First, since we'll be talking about oscillations, I'd like to take a moment and ask, what are oscillations? Well, an oscillation, not just in physics, but in general refers to any kind of repetitive back and forth motion of an object or quantity around a central equilibrium point. It is essentially a cycle of repeating fluctuation, such as a swinging pendulum, a vibrating guitar string or tuning fork, or even the rhythmic hand clapping. Now, there are also electromagnetic oscillations, which are generated by vibrating positive and or negative electrical charges. And these produce oscillations in the electromagnetic field. And you can see on this slide, I put a kind of classic figure of an electromagnetic wave, like radio waves or even frequencies of light. And in biology, we also see oscillations. Biological oscillations are periodic, repeating changes in organisms that fluctuates around stable central values, such as sleep-wake cycles, heartbeats, or even the cell cycle, the cycle of cell division. So you can see we have some brain waves here. We have the lub-dub of a heartbeat in the cardiac cycle. And these kind of cyclic phenomena in living organisms go right down to the cellular and even subcellular scale. Now, in quantum mechanics, we often talk about coherence. So you might be wondering, can oscillations be coherent? Or if you've heard of them, what are coherent oscillations? Well, we see them in classical physics. Basically, the idea that if a signal is coherent, that all the oscillations in it, all the various waves like sine waves, line up. So classical coherence is about oscillations, that is to say waves, keeping in step. The crests and troughs are all lined up and locked in a single color or phase, like the beam of a laser pointer. Now, if we scramble the phases across many colors, it becomes incoherent, like light from the sun or an incandescent bulb. And the ordering is all relative to the waves themselves. So if you look at the main diagram, we see some waves that all line up. They all have the same wavelength, and they're all in phase with the peaks lined up together and the troughs lined up together. But if these waves decohere, so to speak, they become more incoherent. You'll see not only are some waves not lined up, that they might have different wavelengths, and the waves themselves can experience sudden discontinuities. And this is really something that we see ultimately in all light. You don't see perfectly coherent light anywhere, and you don't really see perfectly incoherent light anywhere either. But we do typically see different ranges of coherence. If we have a laser pointer, it produces a highly coherent beam, so coherent it goes in a straight line, a laser beam. If you look at something like an LED bulb, it's fairly coherent, not as coherent as a laser, but still monochromatic light, for example, red, is easier to line up because all the wavelengths are the same length. If you look at something like a white light bulb, now there's automatically already different wavelengths of light. So they're not going to line up perfectly because they can't. They have different lengths. And so you see a variety of different levels of coherence, not just in light, but in many areas of the, ultimately nature. So what is quantum coherence? Well, in quantum coherence, the phase agreement is between probabilities. A quantum system can be in a superposition of several states at once, and the coherence is now the phase relationship between those different states, those different probabilities that we measure the system in a particular state. And so this is a basic introduction to the idea of coherence because this will be significant as we proceed in the book. So we are going to start from the beginning, oscillations in cell structures. Of course, all living things keep time in some way or another. Circadian rhythms turn over each day. Cell cycles take hours. Calcium waves in the cell might last for seconds. Neural action potentials can fire in milliseconds, and molecular vibrations can be even shorter, on the order of picoseconds. So biology's clocks are a feedback loop that keep biological time. Now, if we look into a cell, a eukaryotic cell, zooming in a bit closer, we're going to see the nucleus and various cell organisms, and especially the cytoskeleton, the structural skeleton of the cell. Now, it's not static. In fact, the skeleton of the cell is a highly dynamic entity that undergoes constant cycles of assembly and disassembly, and it exhibits rhythmic behaviors across several scales. For example, the microtubules in cilia and flagella engage in rhythmic beating. Actomyosin in the cytoskeleton produces waves that govern cell motion and shape. And microtubule electrical oscillations, microtubules being the main structural component of the cytoskeleton, have been correlated with neuronal signaling and brain function. So if we zoom in a bit closer to the main structural components of the cell, of the cytoskeleton, the microtubules themselves, microtubule oscillations can refer to both the structural assembly and disassembly cycles and spontaneous electrical signals in them. In cells, microtubule dynamics can occur across multiple frequencies, driving cellular scaffolding and transmitting electrical impulses that are increasingly linked to neural communication, cognitive function, intracellular communication, and even potentially biocomputation. And you can read Mosin's 2025 thesis on this topic. So what are microtubules really? You can see here in this lovely fluorescence microscope image, the microtubules of the cell have been stained in green. The actin is in orange. These are the two main components of the cytoskeleton of the cell. And the nuclei, of course, are very recognizable in blue. The cytoskeleton plays a multitude of roles in the cell, both provides structure, of mobility, allows for transport, and the microtubes themselves, as the main structural component, help enable all these aspects of it. The cell's movement, as I mentioned, transport, as well as cell division, mitosis, and meiosis. And the microtubules accomplish all this alongside actin and intermediate filaments, as well as a number of other components as well. Now, the microtubules themselves are built from tubulin. They're made from alpha and beta tubulin monomers that form dimers arranged in polar tubular structures that exhibit vibrant dynamics. Microtubules are particularly well known for their role in neurology because these microtubule networks, microtubule bundles, in fact, give shape to neurons in the nervous system, in the brain. Microtubule ordering is essential for growth, learning, and memory, and cytoskeletal dysregulation is linked to cancer and neurodevelopmental disorders. So the cytoskeleton is very important for the health of the cell and the health of the whole organism as well. So a lot is known about microtubule, both in terms of their structure and dynamics. Without going into a lot of detail on this slide, many parameters have been measured. As I mentioned, these are very dynamic structures, so we've measured their growth rate, their shrinkage rate, the associated chemical rate constants, the free energy of GTP hydrolysis that drives these dynamics. They can exhibit a pushing force and a pulling force, and as a microtubule undergoes its dynamics floating in the cell or in a test tube, it has a certain persistence length. The list goes on, but the idea is that the microtubule are built of these tubulin monomers that combine to form dimers. You can see a pair of dimers in the center of the slide. The dimers tend to come preloaded with the cellular currency GTP, similar to ATP, if you've heard of that. And as the microtubule grows and transforms dynamically in time, the GTP is hydrolyzed into GDP. I'll talk a bit more about that in a moment. You can also see the roughly the width of the microtubule here on this slide, and I'm very grateful for the individual who posted this figure on Wikipedia because, as you can see, it's a wealth of information and references, so please feel free to look that up. Now, what really happens as the microtubule grows? So GTP is guanosine 5-triphosphate. This energy currency molecule of the cell drives microtubule polymerization and produces microtubule growth. But growth can eventually falter as the GTP hydrolyzes to GDP, and catastrophe can ensue as the microtubule falls apart. But this is not necessarily a permanent disaster, as rescue can occur and the microtubule growth resumes. So if we take a look at this beautiful computer graphic rendering that you can find at the linked site on YouTube, you see a bunch of tubulin dimers floating around. Here's one of them. It's about 8 nanometers long, and it has both alpha and beta tubulin segments. And if we zoom out a little bit, we can see these alpha beta tubulin dimers forming into microtubule filaments, and those individual microtubule filaments combining and kind of zipping up, as you can see here, to create the microtubule. As we scroll along the microtubule, we see in this rapid action video the tubulin dimers forming the end cap of the microtubule as it grows and zips up. Now, as you see the little blue splotches appearing, these represent the hydrolysis of GTP into GDP. Now, this changes the conformational shape of the dimer so that as the hydrolysis occurs, the curvature of the microtubule is no longer preferred, and a horn-like shape becomes preferred that ultimately pulls the microtubule apart. So it's kind of designed to fall apart on a certain time scale, but even if that happens in real time, the microtubule can begin to grow again if GTP-laced tubulin begin to attach again. And so we have microtubule oscillation fluctuations between growth and catastrophe that can go back and forth. In fact, they do go back and forth continually in the cell. So I hope this rather vivid description, depiction of the microtubule dynamic instability process gives you an idea of how vibrant the actual dynamics of this system are. And I'm very grateful to my colleagues who have made the catastrophes and rescues visible in this beautiful video rendering. So now you have a sense of the non-equilibrium self-assembly, growth, catastrophe, and rescue process that occurs in every microtubule, in every living cell, as long as the microtubule continues to grow. So microtubules don't just have interesting structural properties, dynamical properties in their growth, catastrophe, and rescue, and they have interesting electrostatic properties as well. Microtubules and, in fact, the underlying tubulin protein are among the most highly charged structures in the cell, and this charge produces large electric dipoles in a proton-rich surface environment that underpin their rich electrical behavior. You can see this figure here I've borrowed from Calra's ACS Nano paper. We see the microtubule with the seam labeled that zips up, and tubulin dimer. And we can see, in addition, the red and blue colored overlay on the structure of the tubulin, the surface representation of the tubulin, that shows negative charged regions in red and positively charged regions in blue. Now, if you look at the spinning figure that I've created on this slide, as it comes around, we can see a very red region. This is the region associated with the so-called C-terminal tail of the microtubule, where most of the negative charge resides on the, tubulin, I should say. And you can look at the structure of this microtubule. You can see the little C-terminal tails actually sticking out of the individual tubulin dimers on the sides of the microtubule shown here. And so, as I mentioned, tubulin itself has an extremely negative charge of about negative charge associated with about 52 electrons at pH of 6.9. As I mentioned, it's one of the most negative of any cellular protein, and nearly half of that charge sits on the flexible C-terminal tails that protrude from the microtubule surface. Tubulin also has a very large electric dipole. Each tubulin dimer has a large permanent dipole, about 2.8 dBi, and oscillating fields can induce giant dipoles up to 340,000 dBi. So it is not just a molecule with interesting electrostatic properties, but very interesting electrodynamic properties as well. And now, of course, all this charge concentrated on the surface of a microtubule, where the C-terminal tails are sticking out, certainly has an interesting effect on the chemical environment surrounding the microtubule. The microtubule's very large negative surface charge raises the local proton concentration near the microtubule by a factor of 10, lowering the pH relative to that of the bulk solvent. So each microtubule is typically surrounded by kind of a cloud of protons, positive ions that are attracted to its negatively charged surface. So, how does this affect cellular activity? Cells, of course, have their own kind of awareness of their environment. And this is sometimes referred to as basal cognition. Microtubules enable ciliary and flagellar control, axonal transport, and other key aspects of how cells acquire, store, and process information, and ultimately interact with their environment. And this basic ability of the cell to be aware of its environment, for a single-celled organism, for example, it has to search for food or avoid danger. This basic cognition is known as the basal sense. Now, models of nonlinear dynamics have been used to describe how microtubules coordinate motor protein activity and transfer information within the cell based on non-Markovian mathematical models. What does non-Markovian mean? It basically means the system has a memory. It doesn't just instantly forget what happens to it, and that allows it to respond. Now, these memory effects are believed to be linked to electrodynamic effects in the cytoskeleton. And in fact, cytoskeletal proteins were proposed to use nonlinear electrodynamic phenomena to foster biological organization and information processing, in a famous proposal by Hameroff, Smith, and Watt in 1983. Microtubules have also been implicated as having topological order. They have a tube shape, and they have, in fact, the same dimerized, a physicist would say, sublatticed ordered structural motif that generates topological band structure in condensed matter. So what do we mean by sublattice ordered? We just mean that the lattice, the arrangement of tubulin that forms the microtubule, has a sublattice. That in fact, the microtubule is made up of tubulin dimers, and each one of those tubulin dimers has a sublattice very simple one that contains two tubulin monomers. And physicists have taken notice of this, and proposed that it might produce very interesting physics effects known as topological effects in the system. So a phonon model, a classical model, was proposed in 2009 by Prodan and Prodan. They considered a dimerized alpha-beta tubulin sublattice, of course, to begin with, which I just explained, and that forms tubulin fibers that form tubulin sheet, and they modeled it as a spring-loaded lattice. If you imagine rolling a sheet of tubulin dimers up into a microtubule, and they're all attracted to each other, and they can kind of vibrate against each other back and forth. So Prodan and Prodan have proposed that phonon bands can gain a non-zero Chern number that give it interesting topological effects and what are known as protected vibrational edge modes. But the only thing missing from their model that would really give it weight in terms of predictive power is that what's needed is an axial chiral field, or some kind of symmetry breaking term that's necessary in order to produce these interesting topological effects. So most of the physics is there, but someone would need to follow up to explain where this chiral symmetry breaking term could actually come from in real life. And if we could find that, then we would have good reason to believe that the microtubule has an interesting topological phonon band structure. Now, this model was further developed into an electron model, a quantum mechanical model, once again based on the idea that this dimerized alpha-beta sublattice could form alternating bonds on a cylinder, and that the electron bands that form would result in a topological phase, again with protected states that are robust to disorder. But in this case, what is missing from their hypothetical theory is that the alpha and beta tubulin would need to exhibit some kind of degeneracy. That means they're the same. In physics, degeneracy generally refers to energetic degeneracy, which just means two systems have the same energy. In this case, alpha and beta tubulin are not identical, so it's not obvious how that degeneracy would occur in real life, but these are nevertheless very interesting and important models that will become particularly important if the missing ingredients, so to speak, the symmetry breaking term in Prodan and Prodan's classical phonon model, or the necessary coherent intertubulin hopping at the degenerate level in Subramanian's proposal from 2023. So at this point in time, no topological edge mode has been detected in a microtubule yet, but further research is necessary to see how those missing ingredients might be introduced to the microtubule system, either in an experimental laboratory condition or perhaps even in real life in the highly dynamic environment of the cell. So let's get back to these electromagnetic oscillations in microtubules. As I've discussed, microtubules have very interesting mechanically dynamic properties, and their electrical properties and their electrostatic properties also suggest very interesting electrodynamic properties as well. Microtubules reorient in the presence of electric fields due to their large electric dipoles, and they can increase the conductance in an ionic solution. Of course, you get all these positive ions forming along the surface of the microtubules, and it creates an area where they can just slide along the surface of the microtubule, and naturally the system conducts better that way. Now, what's interesting is that an AC voltage applied over a bundle of microtubules bathed in a salt solution has a pinched hysteresis curve in its voltage current plot. And that tells us that the lattice, the microtubule lattice itself, stores memory of the field. And this presents the possibility that the microtubule itself could be used as basically an electronic component. It doesn't just resist current flow on a voltage current plot, it has some memory of what the current was in the past, and that's what we call a hysteresis curve in electromagnetism. And so this would give it properties not just of a resistor that resists current flow when a voltage is applied, but what's called a memristor. It's a resistor that has some memory of the current that's come through it, which makes it very interesting as a potential information processing device, like any electronic device we see in electronics today. Now, it's been proposed that microtubules also can exhibit a cavity enhancement in the presence of oscillating electromagnetic fields that in fact enhance microtubule polymerization rates due to the presence of that electromagnetic pumping from the AC field. This has been proposed to drive tubulin assembly at specific resonance frequencies, even in the absence of GTP, to drive microtubule growth or stabilize the growing tip. Now, that is particularly interesting because the idea is that the oscillating electromagnetic field itself can drive microtubule polymerization. And there were some very interesting experiments performed by Sahu and co-workers in 2014 to demonstrate results to this effect. Now, this is actually not that surprising because electromagnetic fields have also been known to disrupt cellular division, to disrupt the microtubule's role in cell division, which of course is very important to form the spindles that actually pull the two daughter cells apart during cell division. Alternating electric fields can disrupt microtubule polymerization in replicating cells as the spindle misassembles, that is to say, assembles incorrectly, and the cell dies. And the use of alternating fields to drive the polar structures, such as the microtubules, in the cells has become the basis for an improved cancer therapy, effectively killing cancer cells as a result of this electromagnetic interaction between the microtubules, the spindles, and the field itself. So you can read more about that in Person, 2004, and if I recall correctly, the FDA approved this treatment for certain forms of cancer in 2015. So how do electrical oscillations, microtubule growth effects fit together, and could there be an innate cavity effect in the microtubule themselves? So we know that GTP hydrolysis drives, in some sense, microtubule assembly and growth, as well as another process known as treadmilling, where the tubulin basically falls off the back end of the microtubule, picks up a GTP somewhere along the way, and then reforms at the front of the microtubule. So the microtubule kind of grows and treadmills through space. Nearly all of the energy from GTP hydrolysis is stored mechanically in the microtubule lattice rather than being released as heat. So we know we have a non-equilibrium system here that can store energy in its structure and possibly in its vibrations. Axial vibrations were proposed to produce an oscillating electric field in microtubules, of course, because microtubules are electrically polar. So if the system is dynamic, it's out of equilibrium, and is vibrating, it would also be expected to produce an electric field. And in fact, based on the evidence I presented on the previous slide and additional studies that showed that terahertz radiation can cause microtubules to disassemble via a non-thermal coupling mechanism that likely couples to the oscillatory dynamics, we have a pretty strong indicator that microtubules are electrodynamic structures as well. Now, what about this question of whether there's a cavity enhancement? Resonance measurements have shown that microtubules' internal atomic water channel may behave as an optical cavity. Now, this is very preliminary, but the idea is that it could condense pumped energy into discrete levels and couple polarons. Those are quantum mechanical polar quanta to the electromagnetic field as cavity modes. Now, this idea is very preliminary, but it certainly presents the idea that in addition to storing energy and interacting with electric fields, it could actually store energy in the field of the microtubule themselves. Now, this raises the question of the index of refraction in microtubules, because usually cavity needs to, in quantum optics, capture light either using mirrors or total internal reflection in some kind of medium with a high refractive index. And indeed, some studies have shown that microtubules under certain conditions could have a refractive index as high as a diamond. Now, that's a bit surprising because dielectric theory only predicts that microtubules should have index of refraction about 1.5 to 1.7, which would be much more normal, not the kind of thing that's going to confuse you with a gemstone, for example. And in fact, critical angle and reflectometry experiments have affirmed that estimate, predicting a refractive index between 1.6 and 1.9, perhaps a little higher than that predicted by dielectric theory, but not as large as something like a very sparkly object like a diamond with a refractive index well above 2. So I think a little further work needs to be done on this. I've speculated in Physical Principles of Quantum Biology that there could be a plasmonic effect at play here, enhancing the microtubule's apparent refractive index. But at this point, the experimental work does not clearly support that hypothesis, and it would be nice to settle what is microtubules' actual index of refraction, both under normal conditions and under more exotic conditions when it's being driven by polarons or plasmons in this kind of case. Nevertheless, nanophotonic and cavity QED effects have been proposed in microtubules, and a variety of scientific studies have suggested that microtubules may behave as coherent systems that can store and trap light, similar to a fiber optic cable. And in fact, that they might even be optomechanical systems that can create interactions between light and the mechanical modes of the microtubules. So in my own work, for example, the microtubule tryptophan networks were shown to form superradiant states that can increase the fluorescence quantum yields that were measured in our experiments using steady state spectroscopy. So we have a good reason to believe that coherent light matter interactions do exist in microtubules. And in fact, in some experiments that also supported this view, triplet excitons migrated up to 6.6 nanometers in microtubules tryptophan networks in realistic physiological conditions that significantly outperformed the migration rates that would be expected based on standard classical limits. So they really do seem to be able to store and coherently transport excitations of the electronic subsystems that would be produced by the absorption of a photon. Now, in a much more far-reaching picture of the system, microtubules were proposed for scalable quantum computing. And the idea would be that microtubule interiors, that water network, could be treated as a high-quality quantum electrodynamic cavity, which may support strong interactions. Now, this is a very curious idea, but really the question is, how would you get a high-quality cavity? Either we would need the microtubule itself to have a very high refractive index, or the structured water inside the microtubule to have a very high refractive index. As I've mentioned, there have been some suggestions of this kind, but follow-up work has yet to confirm these kind of predictions. So it's not clear how a microtubule could be a high-quality electrodynamic cavity of the kind that we see in typical quantum optical experiments, or even a strong enough cavity effect to give it the properties of something like a fiber optic cable. This is still up for debate. Nevertheless, coherent quantum effects in microtubules are likely to play some role in their structure because there's been a variety of both theoretical and experimental work that have shown both coherent interactions between the excitons, the excited electronic states of the microtubules, and even coherences between the polar vibrational states and the excited electronic states of the microtubule. And that kind of interaction between the excitonic state and the mechanical vibrating state of the polar microtubule would be necessary in order for the microtubule to act as a optomechanical device, a light system that responds mechanically to light in a way that a physicist would consider interesting. And now we have good reason to believe that microtubules interact with UV light due to this tryptophan fluorescence resonance effect. As I mentioned, terahertz waves can disassemble a microtubule, and even previous work by Albrecht Buehler showed that microtubules, and in fact the centrosome of the cell, appears to act as a kind of cellular eye that can detect and respond to infrared light. So there's a strong body of evidence that say optomechanical effects exist in some form in microtubule, and the question is really to get down to the bottom of these numerous reports. So how could we do that? For example, electronic excitations in microtubule lattices have been predicted to interact coherently with polar phonons, forming exciton-polaron quantum quasi-particles. And this would be the formation of a hybrid state of matter that links an exciton, this excited electronic state, to a polaron, this state of polarization of a mechanical vibrating state. A phonon that is polarized gives you a polaron interacting with an exciton in a coherent way. This immediately gives you the kind of mechanism that you would need to get an optomechanical effect. Now, that is just a prediction. On the other hand, Zhang et al. Have proposed, based on some of their experiments, that the asymmetric Raman line shapes that they observed in their experiments could be interpreted as coupling between phonon modes and a continuum of excitons, once again suggesting the potential for optomechanical control. As once again in my own work, microtubule aromatic networks enable a collective radiant effect known as superradiance, the strength of which was predicted to depend on the network's size, geometry, and the amount of disorder. So this again gives us a potential means of forming an interaction between the state of light, the superradiant interaction, or even superabsorbent interaction between the microtubule and the field of light. And now, if it depends on the size, structure, geometry of the network, this again gives another potential mechanism of control linking the light field to the mechanics of the microtubule. And work continues in this kind of exploratory direction. Lindblad analysis, mathematical technique used in quantum optics, characterized information transfer in microtubule chromophore networks, and identified structural and dynamical factors that could preserve transient non-classical correlations in the system. So there's good reason to believe that there's something going on here and the kind of optomechanical level in this system if we can tease out exactly what are the critical physical features that would enable this. Now, of course, decoherence is an issue. At the beginning of the talk, I described what an oscillation is, what coherent oscillations are, and the critical thing in quantum physics is that the quantum states are coherent. I mentioned something like an exciton-polaron. In order to form a hybrid quantum superposition of an excited electronic state and a polar vibration, a mechanical state, they have to coherently interact. And the question is, how long could quantum coherence last in a living cell? Is it long enough? And is it pervasive enough that this could actually have an interesting effect and actually play into physiology in the way that some people believe that, and scientists have proposed that the electromagnetic, the oscillating electric field in the cell is actually critical for physiological regulation and control. So, estimates of long quantum coherence can last in biological structures like microtubules, may span many orders of magnitude. And people have proposed anything from femtoseconds up to milliseconds, depending on the mechanism and the physical model of decoherence, that is coherence loss. So excitonic, that is electronic coherence, coherence of electronic excitations is expected to be brief, in the range of 1 to 100 femtoseconds, maybe as much as 400 femtoseconds. And these are very comparable estimates to the 100 femtosecond exciton decoherence times that were also proposed in photosynthetic light harvesting systems. Now, is this long enough? Femtoseconds is very short. Even 100 femtoseconds is not a long time. But you have to remember that light travels the length of a cell in a few hundred femtoseconds in a cell that is 10 to 100 microns in length. So for intracellular signaling, it's not unreasonable that actually the coherence effect could have an important signaling role within the lifetime of the coherence itself before it decoheres, so to speak. Any functional coherence must be either very short-lived or somehow continually renewed in the cell's thermal environment, decoherent environment. How would we use that coherence or preserve it through non-equilibrium processes? That is really the question. And in any case, the limiting timescales tell us what kind of coherent processes to look for, what length and timescales that we should be searching within in the cell if we expect these things to actually play a significant role. And of course, the coherence only needs last long enough to be functional. Proposed as a quantum antenna, the microtubule structure in the cell's primary cilium may, for example, shelter coherence from decoherence for use in sensation in a recent proposal just from 2026. So I think there's a lot of very interesting work to be done if we are meticulous in our studies of the decoherence when we propose potential coherent quantum effects as physiologically significant in the cell. So what does this mean for living processes? We all know living things exhibit collective dynamics. I only have to go out my front door in the fall to see flocks of starlings exhibiting collective behavior, making these fascinating patterns as they fly and dart about in a flock in the sky. And collective movements govern many natural and developmental processes from the scale of whole organisms all the way down through tissues and organs right to the cellular level itself. For example, in the laboratory, suspensions of microtubules and kinesin will self-organize into an active nematic that produce large-scale spontaneous flows in a liquid crystalline orientational order that you can see here punctuated by topological defects, places where the flow of the microtubule suspension appears to be broken up. There's a little gap in it, or it turns a corner sharply, and you no longer have a continuous flow of this microtubule suspension of filaments. Now, this is called an active matter system, but the question is how we go from active matter to living matter. How do bioelectric coordination and control play into the actual qualities of living organisms that can respond to their environment, not just actively, but to some extent intelligently? Chemical, electric, and mechanical signals converge in collective cellular decisions, ultimately on the most elementary scale using quantum physics. And I hope I've shown you in this talk today how quantum mechanical coherent effects such as the formation of exciton polarons, exciton-exciton coupling, as in superradiant type effects, and other possible non-Hermitian topologies can play a role in the system. The question of whether something like a microtubule can produce a high-quality optical cavity-like environment, like we see in the laser lab, a quantum optics lab, certainly remains in question. But there are also low-quality cavity-like effects, according to non-Hermitian quantum mechanics, that could even play a role in these systems, even if high-quality cavity-like effects are not achieved. So in conclusion, I think there's a lot of very interesting implications. I've presented microtubules as a prototypical biostructure in which electromagnetic oscillations may play an important role. And I believe they offer a premier testbed for investigating electromagnetic interactions with living matter, and including excitonic, polaronic, even polaritonic, topological, and quantum cavity effects. The question of optomechanical control was raised, as the microtubule lattice couples light to motion, and its mechanical state can thus gate the optical output. There's an interplay between its interaction with light and its actual mechanical motion. And this presents the potential for a mechanism for steering the assembly with resonant electromagnetic fields. Now, in terms of some of the more exotic topics I've considered here today, such as exciton polarons and cavity coupling, microtubules may support optical modes and even dress electronic excitations with polar phonons. And if that is so, the lossy non-Hermitian effects due to environmental couplings could become key features. Unlike the strong optical confinement we see in optical cavities of a quantum optics lab, where high-intensity lasers and high-quality cavities are needed, in fact, in biological systems, we might circumvent the need for traditional quantum optics confinement by invoking lossy non-Hermitian effects instead. And so there are a lot of open questions, and I think the outlook is very exciting because the true scope and functional role of quantum coherence in microtubules and other cellular structures remains at least partly unresolved and requires much more research in quantum biology and biophotonics in order to fully tease out the question of what is going on in these fascinating and highly complex biomolecular systems. So if you've enjoyed today's lecture, please go right ahead and download your guide to quantum biology, Physical Principles of Quantum Biology. You can get your own preprint edition for free from the Physics Archive today. It's available now for researchers, students, and curious minds ready to venture beyond classical biological explanations. And there is an updated version, fully revised edition that is now available to order directly from the publisher, World Scientific, or from any number of booksellers online. Please check it out, and if you've enjoyed it, you can enjoy the updated edition in hard copy as well. So, I'm Dr. B. I've enjoyed presenting to you today. If you have any questions, please feel welcome to email me. You can check out other lectures like this one on my YouTube channel, and of course, check out my website, NathanBabcockPhD.com. Thanks again, and have a wonderful day.